what bioinformatics entails, what it's like, what we can do with it, what we cannot do with it, and the opportunities that we have in future if we venture into bioinformatics. And yesterday, I know Mary took us through databases, what are primary databases, secondary databases, and Samad took us through how to retrieve sequences and some other concepts. Like today, I have two people from my team that will be joining us. Samad, we start from where he ended yesterday, and after that, Mr. Tolu, we also join on the call to take us through how to plot and interpret phylogenetic tree. And those are the basic things we will be doing today by the special grace of God. We're also a team member. Like you said, we are open to collaborations and we keep working. And our focus is making the younger generation to know more about opportunities that are being abounded in genomics as a whole. So, Mr. Ayelabegon Abdulsalamad is on this call. I want you to have the lead now. Samad, over to you.
All right, sir. Thank you very much, sir. Good evening, everyone. I hope we all enjoyed yesterday's class. I'm glad to continue from where I stopped yesterday. So yesterday we treated sequence retrieval and blasting. So today we just take us through some selected bioinformatics analysis. So let me share my screen and let's proceed. All right. I hope you all seeing my screen now. Are you all seeing my screen? Okay.
Yes, we can.
Okay, selected bioinformatics analysis. I've listed down some selected bioinformatics analysis here, and we are going to go through them one after the other. So the first one we're going to be going through is druggability. Druggability is like the ability of an active compound to be formed into a drug, like ability to use an active compound to make a drug. So, before researching on active compound, all these biomolecules, either from plant extracts or any biological elements. So you have to check the druggability. When you turn it to drug, can it pass all the tests the drug is required to pass? So for you to check that, there is a specific server designed for that, which is Swiss admin. So I'll be taking us through how to use the server to predict our druggability parameters. So let's proceed. Let me copy the link and go to my browser to do that. So if I'm too fast or you have any question, please do call my attention. I'll be very glad to go back again. So let me paste this website here and let's check how it is. So this is the interface of the website, Swiss Art Like Nature. So it allows you to predict the pharmacokinetic properties, ADME parameters, something like molecular weight and the likes, solubility and all. So for you to predict your drug likeness here, there is two options. My cursor is here, where I'm hovering here. You can easily draw your structure, like our organic chemistry. You can easily draw your structure if you are very good with organic chemistry and you know how to construct a structure. You can just draw it there. Otherwise, you input a list of smiles. So what do we know as smile? So let me take you to PubChem. Let's use a active biomolecule in NIM3 as an example. So let me go to PubChem. PubChem is used to retrieve biomolecules. So just search PubChem on your system, not PubMed, PubChem please, and click on the first link. So I'm going to make use of azadiractine. It is an extract from NIM3. So azadiractine. So it has already brought a list of it here. So I'm choosing the first one. So this is Gr8t, and these are information about it. This is the CID, like the identifier. If you just input this number on the search box, it's going to bring this. So you have your impact name, this is it, somebody smile and everything. So for you to get the canonical smile, just click on it. So let's wait while it loads. You click on it and scroll down. You see here they give you the structure, the 2D structure, they give it to you. Then chemical safety, is it an irritant, environmental hazard, and the likes, they gave it. Molecular formula, everything, molecular weight, they gave it. So just scroll down and get your canonical smiles. This is it. This is canonical smiles. So you just simply copy it from here. You don't need to type it, keep going forth and back. Just copy it, Ctrl C. Then we go back to our Swiss admin tab and we paste our canonical smile, Ctrl V. So once you paste it, you just click on run and allow it to load. Give it a few seconds. Give it a few seconds, it's going to load. It shouldn't take long at all. Probably 5 seconds, it should be done. Alright, we have our result. So this is what we are talking about. It brought the structure. See the structure here, where I'm hovering currently. That's the structure. The polarity, size, everything. It even brought the smile again. So the first thing we are checking in drug likeness is formula. That's not really necessary, just a description of what we are checking. Then molecular weight. You know our molecular weight. Heavy atoms 51, rotatable bonds is 10, hydrogen bond acceptor is 16, hydrogen bond donor is 3. So we have different of parameters here where they give us the respective figures, the respective values. So we have the solubility too. Like if you convert this into drug and you take it, how soluble will it be? So we have it here too. So different thing is there. So for us to check the pharmacokinetics, see it here, J absorption. How is it easily absorbed? It is low. So definitely this kind of drug will be very hard to be absorbed on its own unless it is influenced by other industrial actions, not really laboratory action. I mean industrial action, probably some additives can make it easily absorbed. We have different pharmacokinetic properties. So drug likeness, we follow rules of some scientists. We have Lipinski rule of 5. That's the first one, and I think the most common one out of them. We have Goose, Baba, Egan, Mwek. So from the Lipinski rule of 5, how many does our bioactive components, bioactive elements violate? You see, it violates number 2. That is molecular weight. And under Lipinski rule of 5, the molecular weight must always be less than 500. But from here we have our molecular weight to be 720. So it violates that rule. So our molecular weight is greater than 500. You understand what I'm saying? But if you are not getting this, please call my attention. Then number of hydrogen acceptor. This is it. This NORO. That's number of hydrogen acceptor is greater than 10. And on the normal basis, according to Lipinski rule, it shouldn't be more than 10. It should be below 10. And from here, from the physicochemical properties here I'm hovering, we have 16 hydrogen acceptors. That is above 10. So it violates the rule. If you check other rules, like other scientists' rule, see different violation. Number 3 violation, molecular weight is 480. This goes decided that, okay, the best weight for an active element to be turned to a drug should be 480. Whereas our compound is more than that. It is 720.71. So definitely from this, we can easily conclude that it will be very hard for us to make a drug from this compound. It will be very hard to make a drug because it violates lots of rules. So definitely you might not see, you might come across a compound that violates just few rules, and those rules are easily manipulatable. Like you can manipulate it. For example, drug delivery system, the drug delivery system with lipid coating or nanoparticle coating, lipid nanoparticle or super nanoparticle. So and retrieve the canonical smile of whatever you are going to check, and you input it, then click on run, and you have your results here. That is very easy to do. Okay. So the next one we are going to be talking about is checking antigenicity, toxicity, and allergenicity. Why do we check antigenicity? Antigenic, is it capable of how do I call it? Is it capable of inducing immune response? Antigenicity. When we are talking about antigenicity, let's say you want to work on vaccine design, something like that. Is your vaccine what's the normal way vaccine work? It works by inducing our sorry, please, someone is disrupting the class. I don't know if you can mute the person.
Please, let's mute a microphone. Okay.
All right. So the meaning of antigenicity is just is it able to induce immune response in the body? So let's say we take vaccine now. Vaccine works by inducing immune response, then fighting the foreign object. So we gain adaptive immune response then. I think you understand. We have innate immune response and we have adaptive immune response, which is initiated by inputting a foreign object in our body. So before you can do this, before you can work on a vaccine, you have to find the antigenic part, the part that's capable of inducing immune response. And how do we do this? We have a website for that. We have lots of websites for that. But preferably, choice, different people, and efficacy, I prefer using Vaxigen. So let me take it to where we check how Vaxigen works. So let me search my Vaxigen. So here is Vaxigen. So let's wait while it is toxic. Whether it can turn to like a toxic substance in the body. So we have to check for that. So you can't just work with anything. You have to check whether that's anything a human being or animal, if they are taking it in, you have to check the toxicity so as not to cause any complication. So to check toxicity, we use ToxinPred. So let me navigate to ToxinPred and see how that works. ToxinPred. So we have our ToxinPred here. This is the interface. So to work on ToxinPred, you click on protein scanning. Click on protein. Then there's a place where you input your protein sequence. So let me make use of let me get a sequence from the ones I've been working with before. I think I taught you how to retrieve sequence yesterday, so you should be able to do that on your own. Let me just avoid wasting our time. Let me take a sequence from my end here. Sorry. So this is a sequence, a very short peptide. It's a very short peptide. Let me make use of it. So I click on Ctrl+C to copy it. Let me go back to my website. So Type or paste protein sequence in single letter code. What do you mean by this? You have to paste your sequence Ctrl V alone. But you might come across a server that will request you to post in faster format. How do you do that? You simply do that by just coming to the back here and adding a greater than sign. That's what we know as faster. If they ask you to paste the faster format, just put that greater than sign. So that's all. But this particular server, Toxin Pred rather, is telling us to input the single letter code, just straight code. So we have to remove the faster format. After inputting the peptide, I just click on run analysis. Very simple. And you wait for the results. See the results here. The prediction is all non-toxin throughout. So this means our peptide, this peptide I just copy and I used, in case I want to use it for probably vaccine or any other thing, it is non-toxic. It won't be toxic to the body or anything taking it. It is non-toxic. So that's how we check for our toxicity. Very simple. So let me check if our vaccine is back. I don't know why. Probably they are maintaining the website. And still, I accessed it today. Don't know. Probably they are maintaining it. So it's not working. So let's proceed to the third one. The third one is allergenicity. You know, being toxic is different from being allergic. So you check whether what you are working with can't induce any like allergen, can't cause any allergen, can't induce any allergen. So and you do that using a server called Alatop. So this is the link here. So let me go to my web and get it. Alatop. Let me open a new tab. So we click on this allergenicity prediction and we wait for it to load. I think probably it is having the same issue with oxygen. I think they should be maintaining it also. Because normally it should have loaded by now. So I think they are maintaining the website. And I've been able to show you what the result is like. I don't know. This is a problem from their head. It's not as a result of data or network. It's from the website. So let's just continue. Probably before we are done, they should be ready. So the third one I'll be showing you today is Patrick server. So Patrick is kind of okay. For example, now I told us about composite server yesterday. So what's composite server? Composite server is a kind of server like combines different resources, like different servers into one. It is able to navigate you through different servers while you're on the server. So let me move to Patrick and show you how it works. So we use Patrick for a lot of things, and I'm going to show you a few out of it today. With Patrick, and before you can make use of Patrick, you have to have an account with them. I already have my account and I've been making use of it. So you can all get your account. So to make use of Patrick, you can use it to get the antimicrobial resistant gene. That's the first thing, Antimicrobial resistance is a very a very big issue currently. Like different microbes are developing resistance currently. So a lot of researches are going on antimicrobial resistance. So, and they are documenting it as they are conducting their research. So if you want to check, okay, what are the genes that are causing this antimicrobial resistance in different microbes, so you can make use of this server to check that. So how do you do that? Once I'm on the server, I'm already logged in. See, it's signed out of server. So you just go to annotation. That's the first thing you do. You go to annotation. After going to annotation, this is how it's going to be like. Then you upload your sequence, like the microbe you are trying to work with. You upload the sequence. Here. So clicking on this, I can easily upload my sequence. See, this is the upload sign. I don't know, probably I can make use of a sequence that I've already dropped here, E. coli, something like this. Or let me get another one myself. Let me just upload from my file. So I select file Then let me find where I kept some sequences. So we are working with E. coli currently. So this is a set of sequences I downloaded on NCBI. This is a E. coli sequence. So just click it and open. See, it's already here. I've already selected it. And I click on Start Upload. So it's uploads, you see? It has already uploaded. So the next thing is, which taxonomy are we working with? We are working with E. coli, Escherichia coli. Oh, sorry, let me, you have to choose this. I almost forgot. You have to choose, like, is it a virus? Is it a bacteriophage? Is it bacteria or archaea? And we are working with E. coli, which is a bacteria. So you select bacteria and you type the bacteria name, E. coli. So let me type it in full, Escherichia coli. This is the name we are working with, but this is telling me it is not valid. So that's it. You have to retype, you have to wait till it brings a drop down menu. Yeah, see. We have to select from the drop down menu. So you choose the Escherichia coli and you have to choose the genus, like the broader classification. You shouldn't go for Escherichia virus P something. Just choose the one with Escherichia alone. Select that. Then your label, your identifier. Okay, once your work is done, how do you want to identify it? So I can easily just type anything I want, probably E. coli 1. So any name you wish, E. coli 1, that's it. Then you choose the output folder. Where do you want your results to be stored in? So here you click on this. So I have a lot of folder already. I can just easily choose any one here already. So I choose this one and I click OK. So that's the folder my results will be saved into on the server. So after that, you just click on annotate. See, submitting annotation job. It's already annotating my job. So what is going to happen with this is it's really I think it takes up to 10 minutes, like the least. Sometimes it takes up to 1 hour to run. So I already have some concluded works here. So I'll show you what the result looks like. So to get your result, to get your active job or the jobs you've already done, you navigate to the upper space here where they put workspace. You click on the workspace. After clicking on the workspace, different options are here. You click on my jobs. So after clicking on my jobs, it brings a lot of jobs where you see all this completed. I've already I ran them myself before. So the recent one that I just did, like the one we are just doing, is this running. It is currently running. So to avoid wasting our time, I will just open one of the one I did before and show you how it looks like. So let me click on this one. You don't click once, you double click. After double clicking, bring something like this. You see, the runtime was 44 minutes 22 seconds. It took a lot of time that to finish. So after double clicking, you click on view here. Please, I hope you guys are following me. Click on view. So let's wait for it to load. So after viewing, I told you previously that Patrick is able to do a lot of work. AMR phenotype, if you want to check the antimicrobial phenotype, you'll be able to check it. Phylogeny, you can get phylogenetic tree. Genome browser, circular view, different things. But currently, we are working with the resistant genes. So we are going for specialty genes. You click on specialty genes here. So click on specialty gene and let's wait for it to load. Why is it taking long? Alright, we have the results here. So these are our results. You can easily download the results here, this place. But for example, you are working on just antimicrobial resistance, you have to filter out that before downloading. So for you to filter, check your right hand side. This is an icon, filters. Just click on it. So after clicking, there should be some menus here. Let's wait for it to load. think this should be a network issue. It should have loaded by now. Sorry, let me retry that. It is taking a lot of time. It doesn't take time at all. I don't know why it is taking time currently. I think it should be network. So let me try using another result I have already. Probably it is the result that is having problem. Also, not really sure. Let me retry the filter. Nothing is coming up. So let me try another one here. Let me double click and view. So after viewing, you click on the specialty genes. So we wait for it to load. I think this should be a network issue. Okay, it's already loaded. So let me click on filter. Yes, this brought what I needed. So you check all these filter values. We have the property here. I think we are working with antibiotic resistance. Yeah. So you can easily just click it. So once you click it, it's going to filter this to only antibiotic resistance. Like you are going to see just antibiotic resistance. If you want to, like these are the genes responsible for antibiotic resistance in the E. coli. This is strain we uploaded, like the sequence we uploaded. These are the genes that are causing antibiotic resistance. These are the genes that are causing it. So another thing you should take note of is you can work on drug targets. Let's say, okay, this particular E. coli has antibiotic resistance in this gene. Okay, if you want to develop another drug, where do you think we should target? Which spot do you think we should target? So we have different genes you can target. So I just unmark this antibiotic resistance so that we'll be able to see just only drug targets. So let's wait for it to load. So you can target a lot of GNC, GYRB, everything. It is all here. So you can easily download this and work on it. So another thing you should take note of. So let me go back to the one we are working on solely, which is antibiotic resistance. So let me go back there. So let me unmark drug target. So this, here it is. Another thing is to take note of is I told you that this is like a composite database. This is not a data it's not really all these database we took you on yesterday. So this is also like a composite. It combines different server resources. You see, you check this source from CAD. CAD is a web server on its own. It's a server on its own. You go to CAD, you can perform this action too. Then on that row, that's another one. And their own their main thing, we are using Patrick, and this is the real Patrick. So their own thing is there. So if you want to work with the results on the server alone, you can easily just mark Patrick alone. So it will filter out other sources. And if you want to work on a broader class, you click on the tree and work on it. So classification, you don't need to work on that. Since if you have a specific target, you have a specific classification you want to work on, you can select. But for a broader work, you leave everything untouched apart from selecting antibiotic resistance and the sources. So you select card, you select arrow, likes, and the next thing you do is just download. Click on download. So if you want to download, you can download as text, CSV. CSV is just like Excel, but preferably just go for this Excel for easy navigation. So I've downloaded this already. Let me open it and see what it looks like. So this is what we have after downloading it. We have this. The same thing we had on the website is the same thing it brought for us here. So we have property, antibiotic resistance. We download only antibiotic resistance. So the sources, we have Patrick, yeah. So that's how it works. And the genome name, different things. So if you want to do further analysis on this, probably okay, which gene occurs the most, which gene is less common, all these little stuff, be able to do it easily using this data. So I think that is all for now about Patrick. Although you can use Patrick for a lot of things that you will get to know when you keep on pressing on bioinformatics part. So you can check the protein structure and everything, but I focus mainly on antibiotic resistance today. So I don't know, oxygen is not loading currently. I think the server is down. So it's not really a big deal. So just like our toxin bread, you input the sequence you want to check the antigenicity and you submit. Same thing with Vaxigen. That's how the interface looks like. So possibly you can try it out at home where they give you, okay, input the sequence, you put your sequence. They also collect sequence with plain format, not faster format. You input your sequence and you submit. There's a threshold. The default threshold there is 0.4. So anything below 0.4 is non-antigenic. It can't induce immune response. So it can't induce immune response. But if it is higher than the threshold, the standard threshold there, which is 0.4, probably 0.41, yeah, it is antigenic. You can make use of it. So allergenicity too, it is the same thing with it. So allergenicity, you don't have to use faster format for that too. You just input your sequence in a plain format, not in faster format. So when you open it in a plain format, you have your results. Get it, whether it's allergen or non-allergen. So non-allergen passed, yeah, we are looking for something that is not an allergen. But it's an allergen, that's out of the game. You can't use it for probably our stuff again. I think this is kind of going, this laptop is going, so let me check if it's going to show. Probably they are maintaining the server. It's kind of slow because normally a laptop, Oxygen, are very fast. It's a very easily navigable website. Yeah, this is the same thing. You see, I told you are going to use a plain test. No need for faster format. So let me make use of what I copied earlier, Ctrl V. So you just click on get the results. So let's wait for it to load and see how the result looks like. I think it's still loading. While loading, let me check whether Vaxigen is also back in service. I don't think. I think this server is kind of having problems. So if you try it at home, like after this class, if you try it, you are going to get either it is allergen or non-allergen. So if it is allergen, you should do away with it. If it is non-allergen, you can make use of it. So the site is not responsive. So I think those are the two things you need to check out on your own after this class. It's very easy. I think this is all I have currently to show. So my colleague will continue showing more analysis you can do with bioinformatics. So let me welcome him to join you. He's going to join you very soon. Thank you for listening. So after the class, if you have any question, you can easily drop it. I'm very glad to answer. All right. Thank you so much.
Abdul Samad, thank you very much for that wonderful presentation from you on drugability, antigenicity, toxicity, and using some bioinformatics tools. Thank you so very much for that. As we expect our next presenter, as I said, our next presenter, if you have any question, just get your question ready and prepare them as we open the chat box, and then you drop your questions on the chat box. Hello, good afternoon. Please, can you hear me? Good afternoon, please.
Yes, we can hear you.
Yeah, good afternoon. My name is Gr8t. Please give me a minute to set up my slide. No, I got it. I told them to be. Can you all see my slides now?
Yes, sir.
Okay, all right, thank you. Like I said earlier, my name is Gr8t, and I'll be taking us on sequence alignment, and phylogenetic analysis. It will be mainly more of an hands-on class, but we just take a brief basic on that. We just go through a basic understanding of what sequence alignment and phylogenetic analysis are. Now, we'll start with sequence alignment. And basically, sequence alignment is from the slide, the technique of analyzing and discovering commonalities between biological sequences. That's what the technique is about here. Discovering commonalities between biological sequences. Basically, it is the ordering of sequences, either two or more sequences side by side, so as to find out the similarities and the differences between them. The similarities and differences basically are either conserved regions or regions of divisions, mutations, or substitutions. And then it helps to tell the evolutionary relationships between the sequences, between the organisms you are working on, or if they are the same organism, the strains you are working on. Now, the similarities and the differences might be functional, structural, or the evolutionary links between them. Now, this is an example of how a sequence alignment looks like. You can see the sequence alignment between the histone H1 gene of a human, a chimp, a mouse, a rat, and a cow. You can see the parts where they are similar and the parts where they are different. Now, the parts where they are similar are known as the conserved region. That is, they are similar across board. Now, the non-conservative regions where my cursor is showing are known as the non-conservative region. The parts that are similar are the conservative regions. Let's move on. Now, some of the uses of sequence alignment is the elimination of functionally important region. Basically, you can tell the parts that are similar among you can tell through sequence alignment the functionalities of the different parts of the sequence. For instance, I recently did a work whereby there was a similarity between a particular region across different organisms that perform a similar function. Now, the rest of the sequences, the rest of the sequence of the base pairs were different, but there was a particular part, there were particular regions that were conserved across board. Now, that tells you that those regions for that particular feature among those screens that are similar, those regions are very important in understanding how in the functionality of that particular feature. Now, it also
used to predict the other structures of protein and RNA, and then it's also used in phylogenetic reconstruction. I will be moving fast because of our time. Now, the types of sequence alignment are the global alignment and the local alignment. Briefly, the global alignment is the type of alignment in which the two sequences, seems to be the two sequences are, let me use the word first to cover to the query is the two sequences are the algorithm tries to make the two sequences cover the same length. For want of a better, I don't want to use the technical term to put it in simple terms. Basically, if the length of the two base pairs are a particular number, say 200, and then the other is about 150. The alignment basically identify similar regions within the sequence. The emphasis in local alignment is not on global coverage, that is covering the entire span of the sequence. It's just on identifying it is just on identifying the regions that are similar between the sequences that you are aligning. Now, for want of to define that global alignment is basically trying to ensure that the sequences you align span the similar length, span the entire length, that there is an alignment across the entire length of the sequences that you are aligning. Now, pairwise alignment is basically, in simple terms, the alignment of just two sequences. And whereas the multiple sequence alignment is alignment of two or more sequences. I'm speeding us up because of our time, because you also have to take an hands-on approach. Now, some of the tools used for sequence alignment are cluster, mapped, stick, coffee, muzzle, and K-align. Basically, these are different algorithms that are designed to perform sequence alignment. Now, some of the applications and the servers that are used are Emboss, Patchy, Galaxy. We can see we can see some of the the home page of these tools. Basically, they are tools, they are servers, bioinformatics servers that you can use, whereby you can input your sequences and then they will perform the analysis for you. Emboss, Patchy, Galaxy. Emboss is an online server whereby you just input your sequence and then it runs the application on their server and it runs the alignment on their server and then sends you the result. You can see that's the Emboss first page, is the Emboss link to the first screenshot. Then the Patrick server is also an online server, works similar to Emboss, but it encompasses more functionalities. But within this functionality, within the several functionalities, is sequence alignment. Is the third page, is the third screenshot. Now, it's followed by Galaxy, which is the second screenshot. Then the Eugene. Galaxy is also an online server. Eugene is a desktop tool. Basically, it runs the alignment using your system's resources. So sometimes it can be slow. It's depending on the scale of the sequences you are using. Then Maps is an online tool. NCBI also has a sequence alignment feature within it, and as long with NCBI, of course, you know that Maps and NCBI are online tools. Mega is a desktop tool. Now, some of the things you might come across, some of the way some of these tools present your alignment is basically by color coding. For the most especially for the desktop tools, it gives the same coloration. You can see through using the picture, the first picture as an example, it gives the same color code to parts or regions that are that are similar across your sequences. Take for example the picture. You can see the parts where there's a red coloration. It starts across from top to bottom. It's telling you that at that point, the same amino acid is occurring at that point from your sequence number 1 to the last sequence. Now, for cluster W using online tools, mainly if you use online tools to do your alignments, they they don't use coloration except if they embed in it a viewer. So what they use is asterisk. The asterisk position is at you will see an asterisk below the parts that are conserved instead of a column. Now then, the column uses indicates conservation between some strongly similar properties. That is probably yes, the amino acid might not be the amino acids might not be the same, but functionality are similar. Then the period, basically when there are a few similarities, but they are not strong enough. Quickly, we move on to phylogenetic. We will see all of these once we do the hands-on, once we work practically. Now, basically, phylogenetic tree is a diagrammatic visualization of the evolutionary relationships between the isolates you are working with. Basically, it uses properties of the isolates like biochemical properties, DNA sequences, physicochemical properties mainly, but the most common one that is used is is the DNA sequence. Because of course you know that the DNA, the sequence is the root of every other property that the organism might have. So it uses the sequence alignment to form an evolutionary tree that talks about the relationship between the isolates you are working with. Now there are different statistical methods used in constructing your phylogenetic tree. You have the maximum likelihood Bayesian method, you have the neighbor joining method, you have the maximum parsimony and the minimum evolution. They all have their advantages and their disadvantages. For instance, for maximum likelihood method, the advantage is that it is very precise. Because it runs the tree multiple times before it brings you the results. Are you getting me? But it takes a lot of resources. It takes time. You need if you are running it on your system, it takes a lot of computing resources, and then it can take a lot of time. Neighbor joining is faster, but it is not really reliable because it doesn't run it as many times. It doesn't run the relationship probability as many times as as maximum likelihood. Then maximum parsimony also has its own advantage and disadvantage. Are you please with me? Can you all hear me? Please don't worry, when we start the practical class, you will get these things better. Now, some of the tools used for constructing phylogenetic tree are Maps. Maps is an online tool. Mega is a desktop tool. Emboss is an online tool. Eugene is a desktop tool. And then Patrick is an online tool. Now, the steps for constructing phylogenetic tree are, of course, getting the sequences, arranging them in faster format. We will do all that, but then joining your multiple sequence alignment, then your construction, then viewing it. Now, the tools for viewing it are Mega, IQ3, ITOR. For the sake of this, of this class, we'll be using ITOR as the interactive tree of life. Then there is Eugene, then there is Fig tree. Now, the applications of phylogenetic trees can be used to identify the ancestry of the sequences. You can tell common ancestry of the sequences, and then it can also be used to tell which isolates are more are closer to each other, and then which of the isolates, if there is an ancestor within the isolates you are working with, it can also tell that. Then it is used in study of species evolution. It is used in understanding mutation of sequences per time. It can also be phylogenetic can also be used as a tool to understand the timeline of the evolution. Then it can also be used to study the geography of epidemics. Basically, what that means is probably you have sequences from different locations. You can use it to tell the relationship between the isolates from the different locations, whether or not they are similar, or whether or not the one from one location is an evolutionary offspring of one from a previous the evolutionary offspring of the isolate from a different location. And it is also used in international law. You can be wondering why. There's an example I like to cite, which is one where Club of doctors working as volunteers, I will not mention the countries, were accused of infecting children with HIV on purpose. Then it was supposed to be a witch hunt, and then they took isolates from those from the kids that were brought that they accused them of infecting with HIV, and then when they ran the phylogeny, they realized that the isolates, the what's it called, the isolates in those kits were dated back to before the doctors came into the country. Are you getting me? So it was what saved those doctors from prosecution and jail term in that country. So it can be used in several ways. Now, let's take an hands-on class. Let's practicalize this. First and foremost, does anyone have a question with what I've explained so far? Does anyone have a question with what I've explained? Okay, let's move on if there are no questions. Please, can you hear me?
We can hear you, sir.
All right.
We can hear you, sir.
All right. Yesterday we talked. You are taking retrieval of sequences and then databases. What we'll be doing today, first and foremost, from the example, from the slides, you can. One of the first steps to take is getting your sequences. Now we'll be downloading our sequences from NCBI. Oh, please, can you still see my screen? No, I don't think so.
No, Mr. Tolu. I'm still seeing application of phylogenic tree.
Okay, I understand that. Okay, I'm sharing. Can you see it now?
Yeah.
New tab. Yes. Now, yeah. So you can also follow along if you can. First and foremost, we're going to the NCBI database. And we'll be using the surface. Let's use a BAX gene. It's a cancer marker. Sorry, we go to gene. Basically, you retrieve the sequences you want to use. Yes, so it doesn't necessarily have to be a gene. It can be an entire isolate. But I want to use a gene because it's smaller, and then it won't take much of our time. The point is for you to understand the steps in which that is used to run the alignments. Now we can see different examples here. We are going to take it one after the other, and then we are going to download the sequences. I believe you are following. Please, at this point, if you have any questions, please let me know so that we won't go too far, so we can easily come back and explain things. I believe you cannot hear me. So yes, we download the sequence. We will download the protein sequence. I'm not using in faster format. That's one. We're going to use about 5. What determines? I'm using BAX. I've picked one for Homo sapien. Let's pick one from animal, from mouse. going to rename this mouse, house mouse. So pick 5 of them. This from, thankfully, let's pick from Gr8t. I don't want to open multiple tabs because I'll have to reshare the screen. Or better still, let me just share the entire window. So I can open multiple tabs and our work can be faster. Just give me a minute. Apologies for the delay, just give me a minute. Yeah. I'm having a little problem with my system, sorry. Let's just move on. It's not giving me what I want. So let's just move on. So now we have 3. I stopped at Gr8t. Let's use sheep. That's BX gene from sheep. We all know that it's a cancer marker. Okay, and then the last one.